Good afternoon, students. In the last lecture, we were discussing about the selection methods in the plant breeding. Okay, there are two methods of the selection which are used in the plant breeding. The first one is the natural selection, and another one is the artificial selection. Natural selection, that means the different environmental factors or the nature itself is the uh, main force which is involved in the selection, while in the artificial selection the human being uh, carry out the selection process for their own benefit. That is, we call it as a artificial selection. Okay, here in plant breeding we are mainly concerned with the artificial selection. There are three types of the artificial selection. The first one is the mass selection, second one is the pure line selection, and third one is the clonal selection. Okay, three methods of the artificial selection are there. First one is mass selection, second one is pure line selection, and third one is the clonal selection. We have discussed in detail the first method, mass selection, in detail in the last lecture. Okay, we have discussed the definition and concept of mass selection, then field technique of the mass selection, advantages and limitations of the mass selection. Okay, the question, the long answer question may be asked for the 20 mark in the theory examination as give and detail account on mass selection. Okay, or it may be asked as what is mean by the mass selection and give the field technique or explain the field technique of the mass selection. Okay, that may be asked for the long answer question. Okay, then the in today's lecture we are going to discuss the second method of artificial selection, and this is pure line selection. Okay, in this lecture we are going to discuss the pure line section. Okay, today's lecture is pure line section. Okay, here the before going for the pure line selection, you must understand the terminology pure line. What is mean by the pure line? Okay, here the pure line, the term pure line in this photograph, you can see the term pure line. It was firstly coined by the Wilhelm Johnson. He was a Danish botanist working on the French bean Phaseolus vulgaris in the 1903. Okay, that may be asked for the MCQ. The term pure line, for the first time, it was coined by the W.L. Johnson in the 1903 while working on the princess variety of the French bean Phaseolus vulgaris. Okay, then what is meant by the pure line? Pure line, it is nothing but a group of plants, all obtained from the single self fertilized homozygous plant. Okay, before discussing the pure line selection, you should understand the terminology pure line. The term pure line, it was firstly coined by the W.L. Johnson, a Danish botanist, in 1903 while working on the French bean Phaseolus vulgaris. He defined the pure line as these are nothing but the group of the plants all obtained from the single self-fertilized homozygous plant. Okay, a group of plants all obtained from the single self-fertilized homozygous plant. The progeny of these single self-fertilized homozygous plants, these are called as the pure line. Okay, then another important thing is that the characters of the pure line. Characters of the pure line, these pure lines, these are the progeny of the single self-fertilized plants, or we can say these are the progeny of the self-pollinated crop. Okay, self-pollinated crop, as these are self-fertilized and homozygous. Okay, we are discussing the characters of pure lines. The pure lines, these are the progeny of the single self fertilized plant. Okay, and these progenies, these are obtained by selfing or the crossing genetically identical plants. Okay, the pure lines, these can be obtained by selfing or crossing genetically identical plants. Okay, that is the first character. Then second one, the pure lines, these are genetically uniform. Genetically uniform, these are genetically identical, these are homozygous. Okay, there's the pure lines, these are all phenotypically identical. As these are genetically identical means all these are phenotypically are also identical, except some minor differences. Because these are some minor differences, these are arised mainly due to the variable environmental conditions. ओके, म्हणजे काय, तर सगळे प्युअर लाईन हे जेनेटिकली आयडेंटिकल असतात. जेनेटिकली आयडेंटिकल आहेत म्हणजे त्याचे एक्सटर्नल कॅरेक्टर्स, त्याचे फिनोटायपिक कॅरेक्टर्स हे सुद्धा सिमिलर असतात. पण काही मायनर डिफरन्सेस असतात आणि हे मायनर डिफरन्सेस कशामुळे अराईज झालेले असतात, तर एन्व्हायरमेंटल कंडिशन्स वेगवेगळ्या असल्यामुळे अराईज झालेले असतात. Okay, all pure lines are genetically uniform and also these are phenotypically similar except some minor differences which are arised mainly due to the variable environmental conditions. Okay, and these pure lines, they may become genetically variable with time due to mechanical mixture, natural hybridization, or the mutation. Okay, these pure line, they may become heterozygous or they may become genetically variable mainly due to the mechanical mixture. Mechanical mixture means what? That is, they get mixed with the or these pure lines, they get pollinated with the pollen grains of the other variety. That is, we called it as a mechanical mixture, or their seeds, these get mixed with the seeds of the another variety. Natural hybridization may happen, and mutation, sudden heritable changes may take place in the pure line, and that's why they become genetically variable. Okay, that is, these are the characters of the pure line. Characters of the pure lines, these are obtained by selfing or by crossing the genetically identical plants. Then the pure lines, these are genetically identical, these are genetically identical, and thus these are phenotypically uniform except some minor differences which are arised due to the variable environmental conditions. Then pure lines may become genetically variable mainly due to the mechanical mixture or natural hybridization, or they may be, or they due to the mutation. Okay, that is, that is about the characters of the pure line. Okay, we are discussing the pure line selection, and before this one, you must know the concept of pure line. We have discussed the definition of pure line and also the characters of the pure line. Okay, then production of pure lines. Okay, how exactly the pure lines are produced? We are discussing the production of pure lines in the self-pollinated crops and also the production of pure lines in the cross-pollinated crops. First of all, how the pure lines are produced in the self-pollinated crops. Okay, as all you know, the self-pollinated crops, that is, these are naturally homozygous, and that's why all these are genetically uniform. Okay, and therefore pure lines occur in the self-pollinated crop naturally. ओके, लक्षात आलं. प्युअर लाईन्स म्हणजे काय, तर प्युअर लाईन म्हणजे काय, तर होमोझायगस आहेत, सेल्फ पॉलिनेटेड क्रॉपच्या जे प्रोजेनी आहेत त्याला आपण प्युअर लाईन्स म्हणतो. ओके, आणि सेल्फ पॉलिनेटेड क्रॉप्स मध्ये मग प्युअर लाईन्सचं प्रोडक्शन कसं करायचं. सेल्फ पॉलिनेटेड क्रॉप नॅचरली होमोझायगस आहेत, म्हणजे जेनेटिकली सिमिलर आहेत. त्याच्यामुळे काय, तर त्याच्यामध्ये नॅचरली प्युअर लाईन्स हे प्रेझेंट असतात. ओनली you have to select the certain types of plants and their progenies you have to grown, and these progenies will be called as the pure lines. Okay, that is how the pure lines are produced in the self-pollinated crops. Okay, pure lines, how these are produced in self-pollinated crops? Self-pollinated crops are naturally homozygous and pure lines occur in them naturally. Okay, then the selection is carried out from the self-pollinated crops and their progenies are grown, and these progenies are referred as the pure lines. Okay, then the second one, how the pure lines are produced in the cross-pollinated crops. Okay, as all you know, the cross-pollinated crops are always heterozygous. Okay, these are always heterozygous, means they show the great genetic variation. Okay, these are genetically variable, and that's why these cross pollinated crops subjected for the self pollination for the number of consecutive generations to obtain the pure lines. Okay, understand that pure lines म्हणजे काय, जे जे युनिफॉर्म आहेत, त्याचं जेनेटिक कॉन्स्टिट्यूशन युनिफॉर्म आहे. ओके, तर क्रॉस पॉलिनेटेड क्रॉप मध्ये जर तुम्हाला प्युअर लाईन्स तयार करायचे असतील तर काय करावं लागेल? क्रॉस पॉलिनेशन हे नॅचरली हेटरोझायगस असतात, त्यामुळे तुम्हाला काय करावं लागेल? अशा प्लांटमध्ये क्रॉस पॉलिनेशन तुम्हाला रिस्ट्रिक्ट करावं लागेल किंवा होऊ द्यावं लागणार नाही, आणि म्हणजे त्याच्यामध्ये ते सेल्फ पॉलिनेशनच होईल. आणि हे असं काय खूप साऱ्या जनरेशन मध्ये तुम्हाला करावं लागेल. आणि मग काय होईल? आफ्टर विच देअर आफ्टर number of consecutive generations, the heterozygosity will decrease and the progeny will become homozygous. Okay, and the progeny will become homozygous. But there is a one limitation while using the pure lines in the cross pollination, is that whenever the cross pollinated crops, these are subjected for the self pollination for number of consecutive generations, due to the self fertilization, it will result in the decline in vigor and the productiveness. It will result in the decline and the productiveness in first few generation. ओके, म्हणजे काय होतं, क्रॉस पॉलिनेटेड क्रॉप मध्ये तुम्ही जर सेल्फ पॉलिनेटेड करायला बघितलं तर काय होऊन जातं, पहिल्या काही जनरेशन मध्ये त्याच्यामध्ये त्याचा इल्ड असेल, त्याचा व्हिगर असेल, काय होऊन जातो तर एकदम कमी होऊन जातो. Okay, and this decline in vigor and productiveness, it is very rapid for first few generations, and due to which it will produce the defective and the abnormal individuals. Okay, it will produce the defective and abnormal individuals. For example, the albino plant. Albino plant means what? Here, their leaves, albino, it is a Greek word that means the white. Here, the leaves are not green in color. That is, these are white in color. Chlorophyll pigmentation or the chlorophyll formation does not takes place in such type of the plant, and due to which the death of the plant occur very soon. Okay, that means whenever the cross pollinated crop, these are subjected for the self pollination for number of consecutive generations, then in first few generation there will be the rapid decline in the vigor and productiveness. Okay, vigor and productiveness. Okay, and there will be the formation of the certain defective plants, certain defective and abnormal individuals. For example, albino plant, then another one dwarf plants. ओके, म्हणजे त्यांची हाईट काय तर एकदम कमी होऊन जाते. ओके, त्याची हाईट एकदम कमी होऊन जाते. ओके, बट आफ्टर 7th to 8th जनरेशन देअर डिक्लाइन कीजेस. ओके, म्हणजे जवळपास 7व्या ते 8व्या जनरेशन मध्ये हा जो डिक्लाइन आहे तो काय तर एकदम कमी होऊन जातो, अँड द सर्व्हायबल प्रोजेनीज आर कॉल्ड ॲज द प्युअर लाईन्स. ओके, survival progenies are called as the pure line, and such pure lines, such obtained pure lines in the cross pollinated crops are called as the inbreds. ओके, लक्षात आलं? इनब्रेड म्हणजे काय, तर हे एक प्रकारचे प्युअर लाईन्सच आहेत, फक्त हे कशामध्ये प्रोड्यूस केलेले तर क्रॉस पॉलिनेटेड क्रॉप्स मध्ये ते प्रोड्यूस केलेले असतात. ओके, दॅट इज हाऊ द प्युअर लाईन्स आर प्रोड्युस्ड इन द cross pollinated crops. Okay, we have discussed the concept of pure line, then the general characteristics of the pure line, and also we have discussed the production of pure lines in the self pollinated crops and the cross pollinated crops. Then we are moving how actually the pure line selection is carried out. Okay, or what is the field technique which is used for the pure line selection. First of all, what is meant by pure line selection? In this case, a large number of the plants are selected from a population of self pollinated crop. Okay, here you can see we have discussed in the last lecture the mass selection. The mass selection, it is the method which is useful for the production of the new improved variety in the self pollinated crop, in the cross pollinated crop, sorry. And today we are discussing the pure line selection, which is the method which is extensively practiced, extensively used to develop the new improved variety in the self pollinated crop. Okay, what is mean by pure line selection? In this case, a large number of the plants are selected from a population of self-pollinated crop. Okay, and these are harvested individually and their individual progenies are evaluated, and the best progeny is released as a pure line variety. Okay, that is we call it as a pure line सिलेक्शन. ओके, मास सिलेक्शन मध्ये आपण काय बघितलं की पहिल्या वर्षामध्ये आपण खूप सारे प्लांट सिलेक्ट करतो, त्याचं हार्वेस्टिंग करतो, त्याचे सीड्स जे जमा झालेले आहेत ते सगळे मिक्स करून टाकतो. ओके, आणि मग त्याच्या प्रोजेनी आपण ग्रो करतो आणि 8व्या इयर मध्ये नवीन व्हरायटी आपण रिलीज करतो. ह्यामध्ये त्याच्यात एकदम अपोजिट आहे. यामध्ये जे प्लांट आपण सिलेक्ट केलेले आहेत त्याचे हार्वेस्टिंग करताना त्याचे सीड्स आपल्याला मिक्स करायचे नाहीयेत. ओके, प्रत्येक प्लांटचे सीड्स काय करायचे तर वेगळे कलेक्ट करून त्याला नेमिंग करून त्याला टॅग लावून तुम्हाला वेगवेगळे ठेवायचे आहेत. इथे सीड्स तुम्हाला मिक्स करायचे नाहीयेत. प्रत्येक प्लांटचे सीड्स तुम्हाला वेगवेगळे ठेवायचे आहेत आणि वेगवेगळ्या पद्धतीने त्याचं सोईंग करून त्याच्या प्रोजेनीस तुम्हाला इव्हॅल्युएट करायच्या. ओके, आणि नंतर सगळ्यात चांगली जी प्रोजेनी परफॉर्म करत असेल ती प्रोजेनी तुम्हाला नवीन व्हरायटी म्हणून रिलीज करायची आहे. लक्षात आलं, मास सिलेक्शन आणि प्युअर लाईन सिलेक्शन वेगवेगळं आहे. मास सिलेक्शन मध्ये तुम्ही सगळे सीड्स कलेक्ट करून मिक्स करून टाकत होतात, पण इथे तुम्हाला सीड्स कुठेही मिक्स करायचे नाहीयेत. ओके, how the pure line selection is carried out? In this case, large number of the plants are selected from the population of the self-pollinated crops, harvested individually, and their individual progenies are evaluated. And the best progeny it is released as pure line variety. Okay, that means we can say the pure line selection is the process of separation of crop population into genetically distinct group, each uniform within itself. Okay, simply we can say the pure line selection is the process of separation of crop population into the genetically distinct group. Each group is uniform within itself. Okay, the pure line selection, it is also referred as the individual plant selection, head to row selection, progeny selection, pedigree selection, single line selection, and inbred selection. Okay, the pure line selection, it is also referred as the individual plant selection, head to row selection, progeny selection, pedigree selection, single line selection, and the inbred selection. Okay, the process it is called as the pure line selection. Okay, or in other word, the pure line selection it can be defined as isolating a desirable homozygous individual, isolating a desirable homozygous individual from mixed population and multiplying the same without contamination to release as a new variety is known as the pure line selection. Okay, I have not mentioned this definition into this slide. But just remember the another definition of the pure line selection, as I am repeating this one: isolating a desirable homozygous individual from the mixed population and multiplying the same without contamination to release as the new variety is known as the pure line selection. Okay, that is how the pure line selection is defined in various ways. Here you can see the diagram which is illustrating the various steps which are involved in the pure line selection. Okay, in the first here you can see this is the farmer's field. Now here Roy's bulk. From this one, that is the bulk is a mixture of types and can be sorted out. Single plants are selected. Here you can see the single plants are selected. The single plants are sown individually and tested for purity. Here you can see the seeds of the single plant. These are sown in the single individual row. Here you, you can see this is a one row. That is, these are the progenies of a single plant. These are second row. These are the progenies of the second plant. Okay, that is how, that is their progenies are evaluated. Then here you can see the promising ones, the promising rows. These are later tested for yield in bigger plots using the local types as a standard for the comparison. Okay, best ones are selected and these are then tested along with the existing superior variety, which is called as the check. Okay, then the yield trials are conducted in the Riots field or on the experimental stations, and then here the improved selection is released as a strain from the research station. Okay, this is the diagram which is illustrating the various steps involved in the pure line selection. Okay, which is also called as the single line selection, individual plant selection, then single plant selection, progeny selection, pedigree selection, inbred selection, and head to row selection. Okay, this is just a outline how the field technique, how the field technique in the pure line selection is practiced. Okay, here we are discussing now the actually how the pure line selection is carried out. Okay, pure line selection, just we have discussed, it is the method of selection which is used in the improvement of self pollinated crop. Okay, pure line selection, it is the method which is generally practiced to improve the self pollinated crop, and it takes about generally a period of 10 to 11 years required to produce a variety. ओके, म्हणजे सेल्फ पॉलिनेटेड क्रॉपच्या इम्प्रूव्हमेंटसाठी हे मेथड यूज केली जाते आणि जवळपास 10 ते 11 वर्ष या मेथडला सेल्फ पॉलिनेटेड क्रॉपमध्ये नवीन व्हरायटी रिलीज करण्यासाठी लागतात. Okay, pure line selection, it is the method which is used to improve the self pollinated crops, and generally it takes about 10 to 11 years to produce a new variety. Okay, in the first year, how, that is a large number of the plants are selected. These are the activities we are discussing which are carried out in the first year. That is, the large number of the plants are selected from the mixed population at the time of harvesting. The seeds are picked from each plant and kept separately and numbered. Okay, this in general, these activities are carried out during the first year. As far as possible, the large number of the plants are selected. Then these are harvested. Their seeds are collected. These seeds are kept separately and numbered. Okay, these are kept, selected, and these are then numbered. Okay, then regarding the source of selection, that is source of selection, generally the selection, it is carried out from the farmer's field or the research farm or the research station. Okay, source of selection, from where the selection is carried out, generally the selection, it is carried out from the farmer's field or the research stations or the research farm. Then second, another important thing, unit of selection. Unit of selection, generally the individual plant. Uh, these are selected, thickenly planted. Generally, the plant selection it is not possible, which are thickenly planted. ओके, म्हणजे जिथं खूप सारे प्लांट एकदम गर्दी करून वाढतात तिथे हे सिलेक्शन करणं अवघड आहे. जे मोकळे मोकळे प्लांट वाढतात त्याच्यामधून एक प्लांट ते सिलेक्ट करायला सोपं जातं. दॅट इज द युनिट ऑफ सिलेक्शन. Okay, then which are the characters which are observed or which are used for the selection. Okay, all you know that is the selection it is carried out depending upon the phenotypic characters or the external characters, the appearance, height, color, size, resistance to diseases, insect, pest, then frost, frost, lodging, maturity, yield, etc. Okay, these are the characters which are used for the selection. Okay, I am repeating it one, like the appearance, height, color, size, resistance, maturity, then resistance to the insect, pest, diseases, drought, winter hardiness. Okay, then maturity, tillering, yield, etc. Okay, and generally the selection it is carried out at the time of harvesting. Okay, at the time of selection of individual plant, yield may be considered. Okay, at the time of the selection, yield it may be considered as the character for selection, but it should not be the only characters, as it is influenced by the environmental variations. ओके, म्हणजे सगळे कॅरेक्टर तुम्हाला कन्सिडर करायचे, फक्त इल्ड हा एकच क्रायटेरिया लक्षात ठेवायचा नाहीये. कारण का, इल्ड हे कॅरेक्टर बऱ्याच वेळेला काय तर एन्व्हायरमेंटल रिलेटेड असतं. त्याच्यामुळं फक्त आणि फक्त इल्ड हा एकच क्रायटेरिया यूज करायचा नाहीये. दॅट इज इन्स्टेड ऑफ दॅट वन, द कॅरेक्टर्स ऑफ द होल प्लांट्स आर इम्पॉर्टंट दॅन दोज ऑफ एनी पर्टिक्युलर पार्ट. Okay, the characters of the whole plant are very important than those of the characters of the particular part. Okay, the more uniform is the plant in all its part, greater is likelihood that they will prove better for the selection. Okay, understand that the plant should be uniform in characters in all the plant parts. Greater is the in the part, and that will be it will prove better for the selection. Okay, then how many number of plants are selected in this method? Okay, that is, as far as possible the large number of the plants should be selected, but it is depending upon the amount of land, funds and facilities available. ओके, किती प्लांट तुम्हाला सिलेक्ट करायचे, जास्तीत जास्त प्लांट सिलेक्ट करा. पण ह्या गोष्टी कशावर डिपेंड आहेत की तुमच्याकडं फील्ड किती अवेलेबल आहे पुढच्या एक्सपेरिमेंटेशनसाठी आणि फंड्स आणि फॅसिलिटीज किती अवेलेबल आहे. Okay, during the first year, the large number of the plants are selected from the mixed population at the time of harvesting. Their seeds are picked from each plant. These seeds are kept separately and numbered. Okay, that is the major difference between the mass selection and the pure line selection. In the mass selection, the plants are harvested, their seeds are collected, these are composited together, mixed together, and these are then used for sowing in the next generation. But opposite to that one, in the pure line selection, the plants are selected individually, then these are harvested individually, their seeds are collected, these are not mixed. The seeds of the each plant, these are kept separately and these are numbered. Okay, these are the activities which are carried out in the pure line selection in the first year. Then in the second year 25 to 30 seeds of each of the plant, these are grown in individual row for the observation. Okay, about 25 to 30 seeds of each of the plant, these are grown in individual row for the ऑब्झर्वेशन इन इंडिव्हिज्युअल रो फॉर द ऑब्झर्वेशन. म्हणजे एका प्लांटचे 25 ते 30 सीड एका वेगळ्या रो मध्ये त्याचं सोईंग केलं जातं ऑब्झर्वेशनसाठी. ओके, म्हणजे एक रो एका प्लांटची 25 ते 30 त्याचे प्रोजेनीज ग्रोन केले जातात. सेकंड इयर 25 to 30 seeds of the each plant. These are collected and these are grown in individual row for the observational purpose. At every 10th row, standard variety or the checks, these are grown for the comparison. ओके, म्हणजे 9 रो झाल्या 9 प्लांटच्या, आणि 10व्या रोला काय, जी सध्याची स्टँडर्ड व्हरायटी आहे त्याचं सोईंग केलं जातं. कशासाठी? तर तुम्हाला त्याचा परफॉर्मन्स कम्पेअर करता यावा त्यासाठी. एव्हरी 10th रो सीडेड टू द स्टँडर्ड व्हरायटी फॉर द comparison. Okay, the rows of undesirable plants are discarded. The rows of the undesirable plants are discarded and the desired progenies are harvested. ओके, म्हणजे ज्याचा परफॉर्मन्स हा त्या एक्झिस्टिंग सुपिरियर व्हरायटी बरोबर कम्पॅरेटिव्हली चांगला आहे त्याच रोज फक्त ठेवल्या जातात आणि बाकीच्या रोज काय केले जातात, बाकीच्या प्रोजेनीज ह्या डिस्कार्ड केल्या जातात किंवा काढून फेकून दिल्या जातात. Okay, the seeds from plants of each row composited together and used as the experimental strain. ओके, म्हणजे समजा 10 रोज आहेत त्याच्यापैकी समजा 6-7 रो काय आपल्याला वाटलं की त्याचा परफॉर्मन्स व्यवस्थित नाहीये, तर 6-7 रो काय केल्या जातात तर फेकून दिल्या जातात. आणि ज्या 2-3 रोज आपल्याला चांगल्या वाटल्या जातात, 2-3 रोज म्हणजे 2 ते 3 प्लांटच्या त्या प्रोजेनी आहेत त्याचे सीड्स कलेक्ट करून एकत्र ठेवले जातात. म्हणजे एका रोज मधल्या प्लांटचे सीड्स एकत्र ठेवले जातात. पहिल्या रोचे सगळे सीड्स एकत्र, दुसऱ्या रोचे सीड एकत्र, असं काय, तर 2 ते 3 स्ट्रेन तयार केले जातात. अँड दीज स्ट्रेन्स आर कॉल्ड ॲज द एक्सपेरिमेंटल स्ट्रेन. Okay, the seeds from the plants of each row, these are composited together and used as the experimental strain. Okay, in the second year the experimental strain is produced. Okay, then in the third year, that is preliminary yield trials conducted by repeating the same process of second year. ओके, सेकंड इयर मध्ये जसं केलेलं आहे की एका प्लांटचे सीड्स एका रो मध्ये स्विंग केले जातात. ओके, आणि नंतर काय केलं जातं, 10व्या रोला एक्झिस्टिंग व्हरायटीचे सीड्स केले जातात, कंपॅरिझन केलं जातं. जे चांगले रोज आहेत तेवढे फक्त ठेवले जातात आणि जे चांगले वाटत नाहीयेत, अनडिझायरेबल आहेत, ते काय केले जातात तर काढून फेकले जातात. इन द थर्ड इयर प्रिलिमिनरी yield trial conducted by repeating the same process of second year. Experimental strain sown in 3 to 4 rows instead of one row. Okay, experimental strain, it is sown in the 3 to 4 rows instead of one row. The desired strain harvested and the seeds collected separately. प्रत्येक रो मधले डिझायर्ड स्ट्रेनचे सीड्स हार्वेस्ट करून सेपरेटली ते कलेक्ट केले जातात. ओके, इन द थर्ड इयर प्रिलिमिनरी yield trials are carried out. Then in the fourth to sixth year, main yield trials are conducted in the fourth to sixth year. Okay, beside yield, observations are recorded as disease resistance, flowering, maturity times, and other characteristics. Okay, then based on these superior progenies identified and selected as the improved strain. Okay, strains which are showing the consistent superior performance over standard variety selected as the improved strain. लक्षात आलं? म्हणजे पहिल्यांदा जे एक्सपेरिमेंटल स्ट्रेन होते ते आता कोणते झालेले आहेत तर ते इम्प्रूव्ड स्ट्रेन झालेले आहेत मेन yield ट्रायल झाल्यानंतर. म्हणजे 4थ्या 5व्या आणि 6व्या जनरेशन मध्ये मेन yield ट्रायल्स कंडक्ट केले जातात आणि जे चांगले परफॉर्मर्स आहेत ते ठेवले जातात आणि त्यांना आपण आता काय म्हणतोय तर इम्प्रूव्ड स्ट्रेन असं म्हणत असतो. ओके, 4th to 6th year main yield trials are conducted. Beside yield, observations are recorded as disease resistance, flowering, maturity times, and other characteristics. And the strains showing the consistent superior performance over the standard variety, these are selected as the improved strain. Okay, then in the seventh year, the seeds of these improved strain are multiplied. ओके, 7व्या जनरेशन मध्ये काय, जे इम्प्रूव्ड स्ट्रेन आहे 6व्या जनरेशन मधले, त्याचं फक्त मल्टिप्लिकेशन केलं जातं, त्याच्या सीड्स मल्टिप्लाय केल्या जातात. इन द सेवन्थ इयर, मल्टिप्लिकेशन ऑफ इम्प्रूव्ड स्ट्रेन इज कॅरीड आउट. Then 8th to 10th year, the district yield trials are carried out. District yield trials, that is generally these are carried out in different experimental stations. Okay, just to check their adaptability to the changing environmental conditions. Okay, and then their performance, it is reported to the plant breeder, and on the basis of comparative performance, one or two strains, these are released as the new variety. Okay, 8th to 10th year, generally, the district yield trials are carried out. That is, the seeds of these improved strains, these are distributed to the progressive farmers in the different regions for the district yield trials. Then comparative yield trials are conducted for 3 years. Their performance, it is reported to the plant breeder in the 10th year, and the breeder, on the basis of their comparative performance, select one or two strains, gives the name, multiply, and distribute the seeds to the farmers. Okay, these are the activities which are carried out in the 8th to 10th year. Okay, understand, I am repeating this one. In the 8th to 10th year, the seeds of the improved strain, these are distributed to the progressive farmers in the different regions for the district yield trials. Comparative yield trials are conducted for 3 years for 8th generation, ninth generation, and the 10th generation. Their performance, it is then reported to the plant breeder, and in the 10th year, in the 10th year, breeder, on the basis of their comparative performance, one or two strains release as a new variety. He gives the name, the multiply the seeds of that strain, and the seeds of that strain, these are provided to the farmers for the commercial cultivation. Okay, this is the field technique which is used for the release of new improved variety in the self-pollinated crops. Okay, it takes about 10 years to release the new variety in the self-pollinated crops. Okay, there are three methods of selection. The first method, mass selection, we have discussed in the last lecture. Mass selection, it is generally practiced for the improvement of the cross pollinated crop, or in other word we can say the mass selection is the method which is used to produce new improved variety in the cross pollinated crop. Then second method of artificial selection, pure line selection, it is method which is used to produce improved variety in the self-pollinated crop. And the third method we will discuss in tomorrow lecture, that is the clonal selection. And clonal selection, it is the method which is used to produce new improved variety in the vegetatively propagating crops. ओके, दॅट मे बी आस्क्ड फॉर द एमसीक्यू. ठीक आहे, एमसीक्यू ला हा प्रश्न विचारू शकतात. कोणता कोणता, हा लक्षात ठेवा. मास सिलेक्शन कशामध्ये यूज केलं जातं? क्रॉस पॉलिनेटेड क्रॉप्स मध्ये. प्युअर लाईन सिलेक्शन हे सेल्फ पॉलिनेटेड क्रॉप मध्ये, आणि क्लोनल सिलेक्शन हे व्हेजिटेटिव्हली प्रोपोगेटेड क्रॉप्स मध्ये यूज केलं जातं. ठीक आहे, आय थिंक दिस इज इनफ फॉर टुडे. Okay, we have discussed in today's lecture the definition and the concept of pure line selection, also the field technique. Okay, then the precautions, merits, demerits of pure line selection we will discuss in detail in tomorrow lecture. Okay, I think this is enough for today's lecture.